Cellular respiration is the process in which cells make ATP by breaking organic compounds. It takes place in the mitochondria and can form up to 38 molecules of ATP. Aerobic respiration is a type of cellular respiration that requires oxygen. The oxygen serves as the final electron acceptor so that electrons can continue being synthesized along the chain. The overall equation for aerobic respiration is glucose plus oxygen makes water, carbon dioxide, and ATP. The first step of aerobic respiration is glycolysis, which yields a net gain of 2 ATP. It can function with or without oxygen. With oxygen, it oxidates glucose to make pyruvic acid, which can later make ATP. Step 1. A new molecule of glucose is converted into a new 6-carbon compound. Step 2. The new 6-carbon compound is split into 2 molecules of PGAL. Step 3. The 2 PGAL molecules are oxidized to produce 2 new 3-carbon compounds. Step 4. The new 3-carbon compounds are converted into 2 molecules of pyruvic acid. The next step of aerobic respiration is fermentation, which requires no oxygen. Depending on the type of fermentation, the pyruvic acid is converted into either lactic acid or ethyl alcohol. The third step of the aerobic respiration is the Krebs cycle, where the cell breaks down acetyl-CoA formed from pyruvic acid in another type of molecule to make hydrogen atoms, carbon dioxide, and some ATP. The Krebs cycle takes place in the mitochondrial matrix. Step 1. Acetyl-CoA combines with oxaloacetic acid to produce citric acid. Step 2. Citric acid releases a CO2 molecule to form a 5-carbon compound. Step 3. The 5-carbon compound releases a CO2 molecule to form a 4-carbon compound. Step 4. The 4-carbon compound is converted into a new 4-carbon compound. Step 5. The new 4-carbon compound is converted back into oxaloacetic acid. After the Krebs cycle, electrons from hydrogen enter the electron transport chain, where they slowly lose energy while they pass through a series of molecules because they are pumping protons from hydrogen atoms into the inner mitochondrial membrane, where a proton gradient is established. This process drives chemiosmosis, the process where ADP and a phosphate group combine to make ATP in the ATP synthase. The ATP synthase makes most of the cell's ATP, or energy. But in some cases, when oxygen is not available to the cell, it has to go through slightly different steps. This is called anaerobic respiration. To generate ATP to sustain itself, the cell must go through fermentation after it goes through glycolysis. There are 2 types of fermentation: alcoholic fermentation and lactate acid fermentation. Alcoholic fermentation is the process by which pyruvic acid is converted to ethyl alcohol, the anaerobic respiration action of yeast and sugars. For example, in the making of wine, it is left to sit for years so the alcohol concentration can become higher. Lactate acid fermentation is the process by which pyruvic acid is converted to lactic acid. For example, lactic acid fermentation is used in the making of cheese and yogurts.